Okay, so yeah, I think I'm a little bit of the mismatched presenter here today. Of course, I'm not an expert in hematology. Last night when we had dinner with the speakers, actually I told them that my experience with hematology was, if I calculated back, 30 years ago, when I was my first internship as a student in the lab of John van Rood in the Netherlands, who I think partially invented HLA subtypes, and it was a very active area at that time. And I still remember it was very exciting, but I still chose to go a different direction. And so this is where it brought me, indeed 30 years later. And I think what I'd like to do today is to give you a little sort of a flavor of where the field is going with respect to these advanced technologies like genome editing, but not without also showing you a few examples how we have used these technologies over the past few years to actually come with novel treatment strategies, identifying new targets and new interesting biology. So the talk is sort of split in two, where I will present some of our older work, where it lays the groundwork for this technology, and then I will spend some time on discussing the newest developments in the field and how I think it will actually advance certainly the build of disease models and the identification of better and novel targets. So, of course, we've been discussing bone marrow transplants today, but I think there's also still a strong interest in what we call pathway target or specific target therapeutics for cancer. And of course, this has been largely driven by the identification of genetic alterations that are present in tumors that could potentially be explored as targets, because we know that these tumors are dependent on the genetic alterations of these so-called driver genes. And of course, in the old days, the pathologist would just distinguish a cancer type like lung cancer in different subtypes like large cell, small cell, squamous. But of course, nowadays, on the right, what you see here is more like a genetic diversification of lung cancer based on the genetic alterations of which we believe are actually the driver events of these cancers. And of course, over the years, many academic efforts and also pharmaceutical efforts have generated small molecule inhibitors that can actually target these molecules or these proteins that are then altered in these cancers, hopefully with the very effective response. And of course, this also has been driven largely by the great success of the BCR-ABL inhibitors, Gleevec or imatinib, that was very successful, of course, in leukemia, and people were really, at that time, really believing that this would be the final cure for cancer if we just understood the driver genes, generate specific inhibitors, and we could actually stop these cancers from growing and surviving. And actually there are quite a few examples around that show that these specific inhibitors really work if you target them to the specific subgroup. So what I point out here is that there's a small group in lung cancer that has these ALK translocations, the EML4 ALK, that activates the ALK kinase. They developed a small molecule inhibitor called crizotinib that inhibits ALK, and you can very nicely see here in this waterfall plot that the majority of the patients that are ALK positive respond to this drug very well. So this is, again, of course, is the same illustration as the Gleevec on the BCR-ABL. You get a very strong response if the genotype is present. The same is true for the BRAF mutant melanoma, where you carry a BRAF mutation. That's also a driver in melanoma. If you make a BRAF mutant specific inhibitor, you get a very nice response. I think this second example is even more interesting because they developed an inhibitor that's specifically targeting the mutant form of this protein and no longer targeting the normal wild-type form that's present in all our cells. So by generating such small molecules, you can increase the therapeutic window because it's unlikely that you're going to target normal cells. So therefore, of course, you can increase the dose. And of course, it has been shown that you need very effective inhibition of this mutant to get a clinical response. So of course, this all looks very good, but of course reality is different, and I've seen a lot of survival plots, so why not showing also survival plots, is indeed that for these targeted agents what we see is initially we see a good response, but then rapidly actually the patients either develop a recurrence or resistant disease, and actually in the case of even BRAF mutant melanoma, you have the standard chemotherapy, dacarbazine, compared to the specific BRAF inhibitor called vemurafenib, and you see initially the patients do better, but overall actually they come down at the same time. So overall survival is not changed, although there's an initial response. And this is not the exception, but also in lung cancer, where we start to treat with a specific inhibitor for the EGFR mutant, also present in lung cancer. Initially there's a good response, but over time, again, we don't really see a big difference with chemotherapy. So there's really a challenge for the application of these kind of small molecules in the clinic. And of course, what we now know is that what we need to get insight in, what is the lack of initial response. Of course, these tumors have intrinsic resistance. And then there's a discordance between the genotype and the targeted drug. Can also be what I showed here, the lack of durable response. Of course, these tumors then develop acquired resistance. They find a way to bypass the inhibition of the specific target. And another thing I think that more recently became really important is that what we see is these late recurrences that we don't really understand. But in part, we think this can be explained by the intratumor heterogeneity, where the mass of the tumor is so large that already resistant variants exist in the tumor before the treatment starts, and they are selected out over time, which can take actually many, many years. So really what we need to do is to find a solution to this problem. So one thing, of course, what we have learned over the years, and especially in melanoma, that when you use these specific inhibitors, we can then analyze these patients and see what kind of resistance mechanisms they have developed. And so what we have analyzed based on patients, you find all these other mutations that they actually acquire. So one thing, of course, is that they can make an alternative form of the BRAF protein that's no longer sensitive to this inhibitor. It bypasses the inhibition of the pathway and the tumors start to grow again. Also other proteins in the same pathway, like the MEK protein, the RAS protein, BRAF amplification, they all can drive resistance to these small molecule inhibitors. And if you see in general, what you see actually in the majority of the cases, we see a reactivation of the pathway that was originally inhibited by these small molecule inhibitors. So tumors, on one hand, you can say maybe they're smart. I don't think they're really particularly smart, but they're with high numbers. And of course, there's just clonal evolution going on, so they can select for that rare variant that has adapted or acquired a mutation that makes them resistant to the treatment. But we now understand this resistant mechanism, so there's pathway reactivation, so you can then also say, well, then you need to treat these patients with something that again blocks the pathway. And of course, this is the idea behind combination therapy, is that we should be able to go beyond this short survival benefit by putting combinations together that makes it more difficult for these cancers to develop resistance. And of course, if we have these combinations, we call them rational combinations, because we need insight in the mechanisms that underlie the resistance, and subsequently then design and develop such a combination therapy. And then of course we can improve response, we can potentially avoid resistance, or at least overcome resistance. If you really understand how these drugs work, what kind of molecular markers are associated with response, you can actually decrease the number of patients that you need in clinical trials because you have a much higher frequency of response in these patients. And we think actually this will be greatly beneficial in the treatment of cancer patients because the trials will be smaller, they will be faster, they will be cheaper, so therefore drugs will enter the clinic at a shorter time frame. Of course, at the same time, if you have this insight, you can also not treat patients which you know a priori that they're not going to respond. So we really believe, and this is also a shared vision, that these kind of combination therapies should be more beneficial than single agent therapy. And of course, there are some examples. What I showed for the BRAF mutant in melanoma, this is single agent treatment. Then you compare it to a combination. Now this is a BRAF inhibitor and a downstream pathway inhibitor called bumetanib. It inhibits the MEK kinase, that's downstream of BRAF. And you already see an improved response. So this is already sort of the first example where we see that these rational, smart combinations can improve response. But of course, it's not simple to just say, okay, so now we know it for all combinations. We have to discover these combinations. So the challenge really is, in this work, is to identify molecular markers that are associated with response, but also molecular markers that are associated with resistance. Then you need the targets that can convert or revert this resistance. And of course, finally, you need these drug combinations that you can use them in a more effective way. And again, all targeting these problems that I highlighted before. So, of course, this is not easy. If you just think about the number of drugs, small molecule inhibitors that we have available for treating cancer patients in excess of 500, maybe even 1,000. So even two-way combinations would be 1,000 times 1,000 divided by 2 is 500,000 combinations, possible combinations of drugs that we could use. Of course, it's a little bit problematic to test this on one patient, let alone test it on all the patients in the world. There will be not enough patients to develop these kind of combinations in this way. So we need a smarter way for that. And of course, this is something that we've been doing, is that we have been over the years developing these, what we call a functional genomic screening platform, as a discovery platform to identify these novel targets. And of course, we started this more than a decade ago by applying this technology. It's called RNA interference, that was originally identified in C. elegans by Greg Mello and Andrew Fire. They got the Nobel Prize for that. that actually you can make a so-called short interfering RNA, and this is in a synthetic way that is introduced into cells, and then apparently mammalian cells, and as model organisms, they have a system called the RNA-induced silencing complex that binds these small RNAs. They are complementary to endogenous RNAs. Then this complex finds the endogenous RNA that's complementary to this short RNA sequence, it binds it, and it can degrade the RNA. So therefore, it silences expression. So it interferes with the RNA by degrading the RNA when specifically recognized. So this was how it was first discovered, and actually in Andrew Fire and Greg Mello, they identified that these small RNAs can be processed from large RNAs in cells. There's a whole machinery that makes this, and there's an important role for these, what they then call microRNAs, that actually they control cell state, cell differentiation, and in many other, even mitosis. So they're very important, but you can hijack this machinery by putting in either synthetic siRNAs or something that we developed is what we call short hairpin RNAs. And now what we make is a short RNA fragment that has a little loop and a complementary sequence, and this hairpin can be introduced into cells, specifically expressed, permanently expressed. It's processed by cleaving this loop off into a small interfering RNA, and it will silence gene transcription. And so now you can actually, for every gene in our genome, all 20,000 of them, we have generated these small hairpin RNA constructs, and we can inactivate every gene one by one. And of course, this now allows us again to attack these questions that I highlighted before. So, and this is something that we've done over the years, and I have one slide in between to sort of give you a flavor of how these screens can work. And actually, so this is something that we developed back in 2004 with the first generation of these large collections where you can inactivate every single gene one by one. But of course, to do these experiments one by one is very labor intensive, so we developed a technology where we can pool all these viruses that encode and hairpin RNA together in one big pool. We infect cells, and then we select them on the specific treatment that we're interested in. And then actually, after they've been exposed to a drug treatment, for example, or not treated, we can compare the relative abundance of each of these vectors in the population by sequencing. And by doing so, what we're more or less doing, we're counting cells that have a specific phenotype. And so if the phenotype of these cells is resistance against the treatment, they will increase in abundance. So there will be more cells that carry that specific shRNA causing by the inactivation of the gene. They can also become depleted from the population, indicating that they induce sensitivity to this treatment. So really by doing one screen, we can interrogate all 20,000 genes for their role in the response to a given drug. And so, of course, this is what we've been doing over the years. I'm not going to go through all the details. So a lot of the screens that we've done are based on proliferative selection. So do the cells grow or not? And do they respond to a drug or not? So if we just skip the top and we go here to the bottom, actually, so here we've done a screen in cells that are sensitive to this ALK inhibitor, crizotinib. So these cells actually carry the ALK translocation, and we treat them, and we look in a screen for genes that can convert resistance to this treatment. And what you see here is that when you knock down this gene, it's called MED12, the cells are completely resistant to this ALK inhibitor. So there are two of them, they're completely resistant. So indicating when you inactivate this gene, MED12, which is part of the mediator complex that controls transcription of many, many genes, that actually if you inactivate this gene, you become resistant. As a control, we can re-express the MED12 protein from a different species. Here we have human cells, and we put in the mouse. This is resistant to the knockdown or the inactivation, and then you restore sensitivity. So this was interesting, but then when we went on for this gene, what we also found that if you take cells that are sensitive to the BRAF inhibitor, and these are then BRAF mutant melanoma cells, also when you knock down this gene, they're fully resistant. If you look at a MEK inhibitor downstream, AZD6244, these cells are sensitive, but if you knock down this gene, they're resistant. So really sort of interesting and intriguing that a single gene, MED12, can drive resistance to many targeted agents. One thing, of course, we have to bear in mind is that this is the ALK kinase, this is the MEP kinase pathway, and they probably crosstalk and interact. So therefore, it's not that surprising that the same gene can cause resistance against both treatments. But then we didn't really understand how this protein, when you knock it down, gives resistance. And because we are not smart, but we can do screens very well, what we actually did is that we did sort of a trick. So we took those resistant cells and we did another screen to resensitize them to the treatment. So now what we're looking for, we have cells that were normally resistant, we retreat, and then we look for genes that when you knock them out or knock them down, will make them sensitive again. So now this would be genes that drop down. And indeed what we found is that we found actually that when we create constructs against TGF-beta receptor, and we knock it down, the cells become sensitive again. So of course this is really surprising, because now we have a single gene that controls the MET12, that controls many, many genes, but we can rescue the phenotype by inactivating this receptor. And I'm not going through all the details, but the TGF-beta receptor is known for its role in preserving state of cells, and particularly the epithelial to mesenchymal-like state. So this is what we call an EMT. And what we observed actually, if we look at gene expression, is that if you knock down the MET12 gene, that you get a signature that looks like an EMT. So the cells switch from one state to the other. So they become more mesenchymal-like, and thereby they become more resistant. And now the problem is that the computer actually sort of messed this a little bit up, but what we've then found, if we look in colorectal cancer patient, we take this signature that's driven by MET12 knockdown, we could actually predict outcome of treatment. And this is an outcome of treatment of chemotherapy. So we know now that the expression of MET12 as a signature for all the genes that it's controlling, what we call a MET12 knockdown signature, can predict the outcome in cancer patients. So, and because it sort of can predict, oh wow, it's all over. Okay, so when it can predict the treatment or the outcome in cancer patients, but what of course we know that most of these patients are treated with chemotherapy. And then actually what we showed is that when you take the same cells, you knock down MET12 and you treat with different types of chemotherapy, although you can't see it anymore, but there used to be here an example where they're highly sensitive to chemotherapy, but if you knock down this protein MET12, you're no longer sensitive to chemotherapy. So you can not only become resistant to ALK inhibitors, to BRAF inhibitors, but also to chemotherapy. And now it becomes interesting, because if you now take this MET12 knockdown signature and you project it on cancer patients and see which patients actually respond to chemo or not. And then actually what you see is that if patients carry a wild type signature, so that's meaning that they don't have this MET12 knockdown signature, they were more or less wild type, there's a separation between the ones that are treated with chemo because they do better than the ones that are untreated. But now if you from this large population select the cells that carry this MET12 knockdown signature, there's no longer a benefit of chemo treatment. So again, what we have here for patients is sort of a transcriptional profile that predicts chemotherapy outcome. And of course, bear in mind that this comes out of a almost in vitro screen, functional genomic screen, where we try to identify these novel targets that predict treatment response or are associated with treatment response. And what's now interesting, and this work has been done quite a while ago, that over the years the field has sort of catched up in this, is what they now have shown that if you look in colorectal cancer patients, that you have now different subtypes of colorectal cancer. So we have an immune subtype, or the MSI, of course microsatellite unstable. You have other subtypes, but one subtype is this CMS4 subtype that's more mesenchymal. And of course, if you remember, the gene that I just described is involved in the transition from an epithelial state to a more mesenchymal state. And now what's surprising is that this mesenchymal state is also called the C subtype. You have the same result as this MET12 signature. So they no longer have a response to chemotherapy. These are resistant tumors. This is the mesenchymal subtype. And even what people have observed, that if you take tumors before treatment, in this case it's radiotherapy, you have the different subtypes. But now after radiotherapy, when there's a recurrence, the tumors actually have switched to the CMS4 subtype. So under treatment, the tumors become more mesenchymal, and as a consequence of that, they become resistant to the treatment. And of course, here it's radiotherapy, here it's chemotherapy, and we would predict that there's many types of therapy that are driven by what we call subtype switching of tumor cells. So again, something that follows really out of the follow-up and validation of these functional genomic screens. Well, a few more examples with that we applied this technology, really getting us closer and closer to the clinic. So I think we have a famous example where we go for BRAF mutant colorectal cancer. And this was an interesting story because this is a tumor now in colorectal cancer that's driven by the BRAF mutation that's identical to the one that's found in melanoma. So at that time, doctors thought like, oh wow, we have a drug for that. We can treat patients with this mutation. This is a genotype-driven therapy with a BRAF inhibitor that's normally also used in melanoma. But actually, if you take these BRAF mutant colorectal cancers and you treat them with the BRAF inhibitor, and actually that's here on the top, now the drug is called Plex, but it's the same as vemurafenib, there's no response whatsoever. And of course, this is what they also saw in the clinic. BRAF mutant colorectal cancer patients are unresponsive to a BRAF inhibitor. So we took this as sort of a clinical need, we built it into a model where we in vitro then took BRAF mutant colorectal cancers, indeed showed that they're unresponsive to the BRAF inhibitor, but then we did the screen again to identify genes that when you inactivate them will make the cells responsive to the BRAF inhibitor. And really to our surprise, we found actually a receptor, which is the epidermal growth factor receptor. When you knock it out, the cells become sensitive to the BRAF inhibitor. And of course, the lucky thing is that we know, again, the EGFR receptor is involved in many cancers, among which is lung cancer. So there are quite a number of drugs out there that inhibit EGFR. And so we have gefitinib and cetuximab. So now we can actually, based on this result, combine, and so now we get to this rational combination, we can combine gefitinib, an EGFR inhibitor, with the BRAF inhibitor in this BRAF mutant colorectal cancers, and what we see is a very strong response. So these are highly synergistic. And this is not only in tissue culture, this is also in a mouse model, a xenograft mouse model, where you see that each treatment alone, the orange and the pink line, but when you put them in combination, you get a very nice tumor control. So indeed indicating that this combination is more effective than the BRAF inhibitor alone, and this is something that you find out of these large-scale screening efforts. So, also I won't discuss what's below here, it follows the same lines, which I think I have to say what's interesting is that here it's BRAF mutant colorectal cancer. We do the same screen in RAS mutant cancers. This can be lung, pancreatic, but also colon, now in combination with the MEK inhibitor. And maybe if you paid attention, I think it's a lot of new things, but the pathway goes from RAF to RAS to MEK. And of course, you would think that this is the same pathway. So that probably the same mechanisms actually act on these tumors. But now to our surprise, we found other genes in the screen. We didn't find EGFR, but we found other receptors, the ERB2 and ERB3 receptor, that are now responsible for the resistance that you find in RAS mutant cancers. So really different combinations. They're difficult to predict, but with these genetic screens, we can identify them. And so now what we know from these pathways, this a global overview, is that there's a lot of what we call negative feedback control. So as soon as you start to inhibit something downstream, it will have consequences on the control upstream in the same pathway. So now what we're doing actually, this is what we call vertical targeting. We're not combining, well, we're combining two drugs in the same pathway, because what I showed you is a RAF inhibitor together with an EGFR inhibitor. So this is two drugs in the same pathway. And this is vertical targeting. These combinations are far more effective. And so nowadays, I don't have the data for you to show, but we actually use triple and quadruple combination targeting within one pathway, and that's highly effective. Of course, the problem with these kind of combinations is to control toxicity in patients, because it's not easy. What's interesting actually is that in these quadruple combinations, we can lower the drug's dose of each individual drug with 90% and still get the same response. So these things are highly synergistic. So if there are adverse toxic side effects due to the chemical entity, this is reduced tremendously. Of course, the adverse side effects from the biological inhibition of the pathway is not reduced. So, and actually, based on these screens, actually, we have now several clinical trials in the hospital and across the world that are sponsored by the big pharma companies that are all based on these combinations that we found with these functional genomic screens. So I think what this illustrates is really that these functional genomic screens, the good thing about it is that they actually are, when you find a gene, they're causally related to the phenotype that you're interested in. So this is far more difficult when you look at gene expression. When you look at mutation, it's more of a puzzle to find those that are really responsible for the phenotype. Whereas if you do these functional genomic screens, you directly hit on a specific protein that has a very important function. Okay, so I think this is historically what we have done. And one of the things, of course, that we noted over the years is that actually what we that from the screens that we have done is that we've found a very low number of validated hits. That meaning that this technology actually is not very good. We can find things, but it's not very good. So what we need is really, we need to improve on these technologies. And while we're in the years that we were improving, not only the technology itself was improved, but of course other genome editing strategies came by. And of course this is the CRISPR-Cas9 system that now has a lot of excitement in the field. So I will spend some time actually sort of showing you what is the CRISPR-Cas9 system and what people are doing with this technology nowadays. So until now, I spoke about RNA interference, which is what we actually call, it's an RNA-guided transcriptional silencing. So you use a small RNA to inactivate the expression of specific genes. The machinery that's used for this system is present in all our cells. So you don't have to reconstitute this system. All you have to do is introduce these short RNAs, and then the system will inactivate gene expression. But over the years, people have also developed other types of genome editing tools that are very either artificial or actually, of course, recently something that they learned from bacteria. So the zinc finger nucleases, those are protein modules that you can generate that specifically recognize DNA sequences. They're fused to a nuclease enzyme that then will cut the DNA. Another way is to generate these talons, or also protein modules that specifically recognize DNA on specific sequences, fused to a nuclease. They also cut the DNA. So although these technologies are very efficient, they're very labor intensive because you have to generate these modules that recognize these specific regions of DNA. Of course, this can be applied, but only in a very small scale, and it cannot be used in a way that we've been using it in these large-scale screening efforts. But then recently, a few years ago, of course, the CRISPR-Cas9 system was discovered, and it was discovered, of course, in bacteria, where now we have almost the same phenomena that was on this side, that we have an RNA-guided system that brings a protein to a specific DNA sequence. Then it will cut DNA. So the major difference here is that it's an RNA-guided RNA cleavage machinery, whereas here we have an RNA-guided DNA cleavage machinery. But of course, because it cleaves the DNA, you have the potential of having a faulty repair, so there can be small deletions or insertions that can disrupt the function of the gene. And this is a permanent disruption in the DNA of the gene. And of course, so these genome editing technologies, they really rely on DNA repair. So what you get, of course, I illustrated for all techniques, you get a double-strand break, a cut in the DNA that has to be repaired by the cell, because cells cannot live with double-strand breaks. So what they normally do is they have two ways of repairing this. This is non-homologous end joining or homology-directed repair. And what's the consequence of non-homologous end joining? This is an imprecise repair machinery. So what will happen when you have a double-strand break, you will start to lose small regions of your DNA. And this can also be insertions, but it can also be small deletions that again disrupt the gene. So you have a permanent inactivation of the gene. But on the other hand, actually, if you provide the cells with a donor template, and normally, of course, if cells encounter a double-strand break in a chromosome, they use the sister chromatid to perform homologous recombination and have a perfect repair. But you can also fool the cell by introducing an exogenous donor template that you can now, after the cut is used for homologous directed recombination, and now you can knock in this fragment D into a gene. So you can now really play with the genome of cells. So, and as I said, actually, this is something that people actually first discovered in bacteria. They described the whole system. This is also where the name comes from. It's called clustered regulatory interspaced short palindromic repeats. And actually, bacteria use this as an immune system to attack viral sequences. So they have sort of a memory. So they have all short pieces of DNA here that are homologous to viral pieces of DNA. So when a virus comes in, they make a short RNA that's recognized by this Cas9 machinery, and it will attack the viral DNA. And of course, when people started to understand this whole mechanism, then they quickly converted it into a technology where you can now also manipulate mammalian cells. But again, this is not normal to mammalian cells. This system has to be introduced completely into cells. So that means you need this RNA fragment, the Cas9 protein, to be able to recognize the DNA and introduce the double-strand break. So, and how does it then work a little bit more in detail? Indeed, is that you have this small RNA fragment that's bound by the Cas9 protein. It recognizes the DNA in specific sites, so it needs for the normal wild-type Cas9 protein, it needs the so-called PAM sequence that directs the Cas9, so it finds this PAM sequence. Then it looks for homology across the RNA, and then when it's there, it will cut on both strands, it will cut the DNA. And then you get this either imprecise repair by non-homologous end joining, or you use a homology arm to knock in a piece of RNA. And of course, now this opens up a whole avenue to start to manipulating cells to build more advanced gene models. So really what we now have is a gene editing platform that allow us to generate these small deletions. And of course, what happens with small deletions is that the reading frame goes out of frame. You no longer make a full-length protein, so you get truncations or full inactivation of the gene. So you can really model loss-of-function mutations that are permanently and stable in cells. Now, the other thing, of course, what I discussed, you can use a small donor RNA to introduce specific mutations. So it uses the template to introduce specific mutations. So now we have a very precise mutagenesis, and you can actually model gain-of-function mutations. And again, I have to stress again, is that this is what we call a scarless technology. So after genome engineering of these cells, you cannot recognize that there has anything happened at the locus. The locus is completely normal. And of course, this in contrast to all other types of genome editing technologies where we introduce viruses, where there's integration of viruses, this all comes with a tremendous risk that viruses will land somewhere in your genome where they have detrimental effects. This technology is completely clean. And of course, it's also not restricted to very small genetic alterations, but you can even put in very large fragments. So you can put large fragments in specific areas of DNA. And for example, what we do a lot is that we put fluorescence reporters behind genes, so we can actually follow endogenous gene expression by fluorescence reporters. So again, so you can remove whole arms of chromosomes, very large pieces, but you can also introduce chromosomal rearrangements. This is something that we could never do. We could never generate a chromosomal rearrangement. But now with this technology, if you design different CRISPRs, so different guide RNAs on different chromosomes, they will each cut, there will be a double-strand break, and then they will fuse the different chromosome fragments. So we can now actually mimic disease that are based on chromosomal translocations, something that was very difficult in the old days. And of course, this is not limited to coding regions, because you can also manipulate other DNA binding elements. So these are, of course, are elements that are used to loop the DNA to generate transcriptional activity of different genes. And now you can actually revert these kind of elements and disturb on a larger scale gene transcription, but also understand how these enhancer elements actually control gene transcription. And of course, there's a strong interest in what we call epigenetics of tumor cells to also pursue this as a potential target for therapy. So what's interesting, and I think you're still with me, sharing the excitement about this technology, although they're all leaving, that they have to catch a plane, I know. That actually, so what we have is a system where you can sort of very specifically direct a protein to a position in your genome, which by itself, of course, is quite surprising because we have a huge genome. We're talking about 3 billion base pairs, where you can specifically locate a protein. So people have continued to think about potential applications of this system. So you can actually also bring this protein to a specific region in your DNA, and now rather than making it cut, you fuse it to other proteins that start to influence the local surrounding of this bound protein. So you can fuse these proteins to transcriptional activators, transcriptional repressors, so now you can turn off the transcription of genes on a transcriptional level. We're no longer manipulating the DNA, but we're using this system to specifically target the protein to a locus. You can even modify the chromatin by fusing this Cas9 that can no longer cut to other proteins that can influence methylation states, acetylation states of protein locally, thereby again affecting transcription. You can fuse the protein to a fluorescent reporter, and now we can actually visualize specific DNA sequences in cells. At the base resolution, at a single base resolution, we can identify individual genes. So, and of course, this is something that people are also using. Is there displacement of proteins on that region? Is there folding? Is there association? So, and this can all be done in live cells. So you don't have to fix the cells. We can follow this over time, dynamically, what's going on in cells. And then finally, a recent switch again. People keep on looking for other proteins. They can do the same. They even found now a Cas9 protein called C2C2 that no longer recognizes DNA, but now the whole system recognizes RNA. So now it can actually cut RNA. Of course, so now we're a little bit back to what I showed you in the beginning, RNA interference. So now we're using a Cas9 protein that recognizes RNA and cuts it. So we have a whole toolbox where we can play around. And of course, that the development in this CRISPR field is going so fast, I think in part is driven by all the knowledge that we gained over the years from RNA interference. So when you look on this timeline, RNA interference was first described in 1998, and then actually it took more than 10 years to get it into humans. What you see here is that the CRISPR system, although it was discovered a long time ago, when it was first introduced in humans actually, now we're already into clinical trials using this kind of technology in patients. So within a few years, people had taken all the knowledge that we acquired over the years to implement with the CRISPR technology to really get it so much faster ahead of the field. So, and I'd like to show you a few examples what we're doing with this CRISPR technology. And of course, this is work of many, many other people that I will show you today is that people using this technology. So one thing, of course, is you can now very specifically generate these disease models. And of course, this is what we do in mice. Rather than manipulating the germline, you can now actually start to genome engineer actually the ES cells and the zygotes before injection. But you can also engineer the mouse itself in the adult tissue, so you can actually put in Cas9 and sgRNA encoding viruses that will infect a different tissue. You can inject plasmids in the bloodstream. They will infect the cells. They will do genome engineering. And of course, what we now also have is a mouse that's transgenic for Cas9. And so now you only have to bring in this RNA, this guide RNA, to direct the genome engineering process. Sometimes I say that we should also start to make humans transgenic for Cas9 for the future. So if you want to manipulate your own genome, it's nice to have Cas9 around already, because this is actually the challenging part of this, to get Cas9 protein in your cells. But of course, we do have it already in mice. They tolerate it very well. There are no adverse side effects. If you put in Cas9 in an adult mouse, without having seen Cas9 before, there is actually an immunological response against the Cas9 protein. So that sometimes makes it difficult. So, but then what people did is that they took this system, and then here's one example actually where people took a mouse and they injected viruses actually that encode guide RNAs that target the major tumor suppressor genes. And when you inject this in a mouse, and this is already a model that carries a KRAS mutation, then what you see, if you put these guide RNAs in the mouse, you see rapid development of tumors. So they go to all tissues and they start to develop tumors. And now what's interesting is that when they can now look at the frequency of the different mutations that are generated by all these different guides in the tumors. And you see heterogeneity across tumors. Some genes are very frequently deleted, so it's APC and the PTEN gene. Other genes are not that frequently deleted. And this is exactly what we see in human tumors. So we start to interrogate the whole spectrum of tumor suppressor genes in one animal model. We can select for the genes that are most important. We are identifying the most relevant targets. Where do we have to focus? So this is really a great advantage of the CRISPR system. Another interesting thing is what they observed in these mice is because they introduce all these different guides at the same time, of course you run the risk that you get chromosomal translocations, because you're targeting different chromosomal arms. And indeed, this is what they saw. They saw a translocation between the PTEN gene and ARID1B. This is what we don't see in normal tumors. We don't see it in humans, but this is something that does appear in these animal models. But as I told you, of course, chromosomal translocations are very interesting for disease. And I told you in the beginning, the translocation with the ALK kinase, the AML4 ALK translocation, we find this in lung cancer. So in the group of Ventura, they generated CRISPR-Cas9 constructs that drive the translocation between EML4 and ALK. So, and then you get in these mice, you get this fusion protein due to the chromosomal translocation, and now you can start to build sort of the human disease model. And indeed, what they do is that you make these guides into an adenovirus backbone. So these are the two guides targeting either the ALK gene or the EML4 together with the Cas9 protein in an adenovirus. You inject them into the lungs, and now they start to recombine, genome edit the cells in the lung, you get the translocation, and you can also get tumor formation. So this really recapitulates the human disease model by putting in these guys, the genome editing reagents into the lungs of these mice. They're responsive to crizotinib, what I showed you in the beginning as well, in lung cancer patients, and now what they also observe is that they develop resistance to crizotinib. So again, we can use this mouse model also to start interrogating what kind of resistance mechanisms exist and what we can actually how we can use this for the treatment of human patients. So I think this is interesting, again, stressing the fact that with these new technologies, we can now build far more advanced human-like tumor models. Of course, so these were tumor models, but I think the applications are not limited to just tumors. This is work that has been done by the Hans Klevers lab, that are in the Hubrecht Institute in Utrecht, where actually they were able to grow these, what they call 3D organoid cultures, from stem cells from the colon. And of course, what they have found is that these, when you isolate stem cells from your colon and you grow them under these specific conditions, they form these organoids. And actually, these organoids, they respond to in a certain way. But what they also found is that if you take these from patients, actually, that have a cystic fibrosis gene mutation, they behave differently than the normal wild-type organoids. So what they show actually is that if you actually normal organoids, actually, if you, I have to see if they swell up or they do not swell up. Yeah, normal organoids, if you give them forskolin, actually they swell up. Whereas if you have a cystic fibrosis mutation, these organoids actually do not swell up. So that's what you see here. If you treat them with forskolin, actually the organoids do not respond. But actually what they then showed is that they can repair the cystic fibrosis mutation by using the CRISPR-Cas9 system. So they make it into a wild-type allele again. And here what they show is that two of these clones independently now indeed start to respond like wild-type cells, that they swell up in this forskolin assay. So I think this, of course, is an example of an ex vivo repair of a single gene, hereditary defect, the cystic fibrosis gene. But of course, you can also use this potentially as an ex vivo treatment and a reimplantation. So and then we're getting more back to the transplantation strategy. So, of course, then the next step is, can we also do this restoration of a disease model in vivo? And of course, one of the popular disease models is for Duchenne muscular dystrophy, where they also now use a mouse that carries the mutation, and then we're trying to repair the mutation by introducing the CRISPR-Cas9 system. So what they actually do in this mouse model, they introduce adenovirus-associated viruses that encode the Cas9 and the guide RNA that actually cut the gene for Duchenne muscular dystrophy in a position where there's the mutation with an aberrant stop. They remove this exon, and now you get a skipped exon, and you make sort of a partial functional protein. So people already knew that this could be done with this exon skipping, but now what we're doing is we inject the mouse either intraperitoneally, intramuscular, or behind the eye. And actually, if you introduce these viruses in this way, what you see actually restoration of this protein into the muscle cells. And they can actually really restore the muscle function in these mice by having these injections of these viruses that will start to manipulate the gene of Duchenne and restoring the wild-type function. So this is now an in vivo application where, of course, you use viruses to deliver the whole Cas9 system. But of course, there's a strong sort of attention to, can we find other ways of introducing these kind of reagents into the human body and thereby, again, causing genome editing and restoring disease. And this is really the group of MIT from Anderson that's doing a lot of this work. And so they really have a non-viral delivery of CRISPR components that can also restore disease. So this is actually a mouse model for hereditary tyrosinemia, where there's also a specific mutation in the FHA gene with a G2A mutation exon 8. And so what they actually do here is they provide, in this model, they provide the guide RNA, the Cas9 protein, but also a DNA template to repair the mutation. So this is all put into these mice. And of course, because they inject this in the liver, so you have to restore this mutation in the liver to restore the disease phenotype. And they can really show that by doing this in mice, where you actually now hit not all, but the majority of the liver cells with this system where you actually have a nanoparticle encoding Cas9, you can restore the expression of the FHA gene in these livers of these mice and also actually restore the disease phenotype that you can observe in this mouse model. So again, indicating different strategies for in vivo genome engineering in a mouse model that's now possible. And of course, if it's possible in mice, we should go the next step and also make it possible in humans. So of course, this would also then be either different strategies, so you can take cells out, do expansion, repair the defect, and then put it back. So this can be done, of course, with adult stem cells. You can reprogram cells, it was just mentioned, take the cells out, fibroblasts, make induced pluripotent stem cells, do a restoration, a repair, and then give the cells back, or actually through a viral delivery of all these components into the human body in specific organs to also repair these disease phenotypes. And I think this is something that we will see in the coming years, that there will be, again, improvement of these technologies to make this possible. So one thing that's very close to us here today, of course, is using CAR T cells to actually as a therapy. And of course, one of the risks continuously with CAR T cells is that you have to introduce the chimeric receptor in some way. It has to integrate in the genome of these T cells, which is a risky undertaking because it can integrate in a position, again, where it has detrimental effects. So what people have now set up is a system where you actually knock in the chimeric receptor in the T cell receptor locus itself. So you're now just replacing the T cell receptor with a different one without disturbing any other element in these cells. So again, I would say is that this is a scarless approach. You won't recognize any of these changes that have been going on except for the knock-in of this receptor. And of course, because it's in the endogenous locus, all the controls are the same. There's no disturbance of expression of other types of control. And so what they show actually is that if you do this in this way, rather than just introducing it by a viral infection, you get a much better response in these animals for the control of disease with these genome-engineered CAR T cells. And of course, in this paper, they describe many of the other features of these cells, that they're much more active, they're better primed, so they really show that this is an excellent way of doing this. And again, so highlighting that it minimizes the risks of insertional oncogenesis, so activating other genes, autoimmunity or alloreactivity, because you don't have any other elements than the reconstituted T cell receptor. There's constant expression, there's no position effects that can create variation among the different populations. And of course, they show actually that it's more potent, generating more potent T cells by delaying also T cell exhaustion. So I think this is again something that we will see over the coming years, that these kind of technologies create a cleaner, more efficient way of manipulating these cells. So of course, there are still some practical barriers that we have to take. So one of the things, of course, is that at this moment, we don't have the capability to identify and enrich for cells that have the desired genome editing event. So we don't really have selective ways of doing this. Of course, the CAR T cells are a little bit different because when you have an antibody against the chimeric receptor, you can purify those cells that you're interested in. Of course, for other types of repairs, we don't have this. You need a almost in vivo functional selection mechanism. Of course, the other thing is that I pointed out already is that there's a requirement of transient nuclease expression, because this is one thing, it can be immunogenicity, causing immunogenicity. On the other hand, where there's continuous presence of the Cas9 protein, it can also start to cut the DNA in other regions and thereby creating off-target effects. And then we're back to square one, because then we have unpredictable genome editing in these cells. Of course, there's also still a challenge to find the good vector systems to deliver the nuclease and the recombination template. This is really challenging. And of course, every time when you generate cells that are going back into the patients, you have to make sure that there's no off-target gene editing events. And this is still quite a challenge in the field to find the good ways of analyzing these populations of cells, whether this has taken place or not. So that's something I think that we have to improve on in the coming years. In the last few minutes, I'd like to go back. I think I have a few more minutes. 1 minute. I'd like to go back to a few. So screening efforts again. The good thing about this technology, because it's much more potent, much more effective and more simple, we can also do these what we call in vivo screens. I showed you in the beginning a few examples that were all tissue culture models. But now actually we can do the same kind of screens in animals. So we infect large populations of cells with many different of these guide RNAs. We select the cells in vivo. And you can select for metastatic capacity, treatment response, just tumor growth, tumor initiation. So all kinds of phenotype can now be addressed actually in vivo in the context of the normal or the diseased animal. The other thing is that I also pointed out a little bit is that because of this technology, we can now also actually screen for phenotypes that are not just based on growth, but also on other. We can put in fluorescent reporters using the CRISPR-Cas9. We can put in cell surface markers, but I think what's also interesting, we can now look for RNA expression. We can do single cell sequencing, where we actually, rather than looking at pools of cells, after the selection with the CRISPR introduction, we can do single cell sequencing and measure per cell the mRNA expression. So this again opens up a whole new avenue for different phenotypes that are really associated to what I call this cell state analysis, which appears to be very important in drug response. Another concept that people are exploring is that they put in different guide RNAs against 2 genes in one vector. So that means that you can create a large population of cells where you inactivate 2 genes at the same time. And so what we're now generating are these what they call genetic interaction maps, where on a genome-wide scale you can look for all possible genetic interactions. And this has been very powerful in model organisms like yeast. So now we're no longer limited to model organisms. We can actually do the same thing in human cells on a genome-wide scale. So this is something that also will be explored over the future. For simplicity, I will skip these 2 slides. So I think another interesting one, and definitely for people that work on developing new drugs and getting them in the clinic, there's always this concern like, how do patients get resistant? How do they develop resistance? And now here we have, again, another trick in the Cas9 toolbox. So what they have done, they mutated the Cas9 first that it no longer cuts, but now they fused it to another protein called AID. And AID actually is a cytidine deaminase that's normally involved in somatic hypermutation and class switching. And so what happens is this protein, when you bring it close to the DNA, it can increase the mutation frequency with over 1 million times. So now what you're doing by bringing Cas9 to this protein to a specific region in the DNA, you will create many point mutations very close to where the Cas9 has bound. So now if you now know the target of your small molecule inhibitor, you can randomly mutate this gene across the whole reading frame in cells while you give them the treatment of your drug. And so by doing so, you can actually find all the mutations in, for example, here in ABL against imatinib, before you have seen any patient. So already identifying what they call gatekeeper mutations that cause resistance to this drug. And this is an example with imatinib, and this is an example with bortezomib. Okay, so I think the last slide here, and then this is another interesting one, and this is sort of the worry of the field a little bit, is that... Is it me? It's the go-to meeting for you. Okay, we're going to talk. We're in a presentation. Okay, so I think which is very interesting. So actually, people what they have identified a few years ago is that actually this MELK protein was a driver of breast cancer, and so this was like a study that they identified this protein as a driver. So pharmaceutical companies started to make small molecule inhibitors of this protein, and they thought this would block the kinase activity and inhibit proliferation of breast cancer cells. When they showed all the biochemical properties of this protein, they started clinical trials with this inhibitor in human cancer. But now, with the Cas9 technology, we can actually now look whether this protein is really involved in the proliferation of these cells. So what they did, they generated guide RNAs that removed this gene from cells. It's completely lost. Then they looked whether it's a driver of breast cancer. So they did it in breast cancer cells. There's no difference between the wild-type cells and the knockout cells. So if you use a clean system, this target is no longer valid. Then they went one step ahead and they showed actually for the small molecule inhibitor against this MELK kinase, there's no difference whatsoever in response of cells whether they have the MELK kinase or not. So that means whatever effect we see from this small molecule inhibitor is not associated to milk whatsoever. So I think, so what we're not only talking here is technology that will give us new targets, but it will also invalidate targets that we have proposed over the years. And I think more than 50% of them actually will not be true when we start to re-investigate them with these very clean type of technologies. So with that, I'm going to stop. I hope that I sort of have convinced you that the Cas9 technology is really a versatile technology to manipulate genomes. You can make all kinds of models. You can do genomic alteration in vitro and importantly in vivo. We're going into the field of as a therapeutic strategy, target validation, and of course what I show, and this is mostly the focus of my lab, as an effective screening tool. And with that, I think I'm a little bit over time and I'm more than welcome to answer any questions. Thank you.